Hello and welcome back to my channel. And today we are going to start a topic that is gram staining. And if you are new to my channels, go to subscribe button and get it subscribed, and also like and share the videos. Okay, so let's get started with this topic, that is gram staining. Okay, so let me change the color of the pen. Yeah, so gram staining was given by Hans Christian Gram. Now you can easily understand that why the staining procedure is known as gram staining, because the scientist's name contains gram. His name is Gram. Okay, it's Hans Christian Gram. That's why the staining procedure is known as gram staining. Okay, and if a bacteria, this staining was given this staining was done on bacteria. Okay, so based on that we we used to say gram positive bacteria, gram negative bacteria. So today we are going to discuss what is the difference between gram positive and gram negative bacteria. Okay, so if a bacteria gets stained by this procedure, we call that as a gram positive bacteria. Means if bacteria gets the stain or this stain through this procedure, it's gram positive, and if not, then it's gram negative. Okay, gram positive and gram negative bacteria. Now let me clear this so I can give you the diagram. Okay, what is the difference between this gram negative, gram positive bacteria according to their structure? Mainly because of their structure, the staining procedure or staining get affected. So if this is gram positive and this is gram negative bacteria. Okay, so if we consider this as the wall, cell membrane of the gram positive bacteria, and outer to it there is a wall that is known as the okay, this outer layer is known as the cell wall. What does it name as? What does it is called as? Cell wall. Okay, which color should I use? Orange. Okay, so this is the cell wall. This portion, this portion is the cell wall, and this cell wall is constituted, or the cell wall is made up of peptidoglycan, which is arranged in layers, many layers in bacteria, and it is rich in acid, that is teichoic acid. Rich in teichoic acid, cell wall. Okay, now this is the gram negative bacteria having cell membrane, and this is the cell wall. Okay, so now you can easily see the difference of cell wall in both the bacterias, gram positive and gram negative bacteria. This is the cell wall. You can easily identify this. Okay, this is the cell wall. It is made up of peptidoglycan, and in this teichoic acid is absent. Teichoic acid is absent. Okay, there is another layer that was found in gram-negative bacteria. It is here like this. Okay, so this is the layer. This is the layer, outer layer of even this is outer to the cell wall. Okay, cell wall, and cell wall is covered by this layer, and this is a glycoproteinaceous layer. Okay, this is a glycoprotein layer which also has polysaccharide and phospholipids. Okay, layer. Okay, and this is the structure of gram-positive and gram-negative bacteria. So now let me quickly write down the steps of staining. Okay, do remember these structures because we are going to get the conclusion from these structures only. Okay, so first step is we are going to fix the bacteria on slide, and that procedure includes the heat fixing. Okay, so first step is heat fix the bacteria on the slide. Okay, second step is we are going to add crystal violet stain here. Okay, after heat fixing we are going to add crystal violet stain. This is about, we are going to leave that stain for 30 seconds to 1 minute. Okay, now next we are going to, after 30 seconds or say 1 minute, if we put that stain for 1 minute on the slide, then after that we are going to wash the slide with tap water or with normal water. Okay, with water. This is our third step, and after that we are going to add the iodine, which we are naming it as a gram iodine. Okay, gram iodine on the slide, and this is also for the 30 seconds to 1 minute time. We are going to leave that gram iodine on the slide for 30 seconds to 1 minute. Okay, and after that, fifth step is we see the slide. In that we see that all the bacteria are purple in color, in appearance. Okay, so after that we are going to wash the slide with 90% of acetone or 90% of alcohol. Okay, we can choose either of them. Now after that, seventh step is that we would find two types of bacteria here. Okay, we would find two types of bacteria. Okay, here, change the color to make it more understandable. Okay, so this is the purple bacteria. Okay, the bacteria which are purple in color. So these are the bacteria which retained the color of the stain. So these are gram positive bacteria. Another are colorless bacteria. For them, for these colorless bacteria, safranin is added. Okay, for these bacteria, safranin is a stain added to them and they appear red after that. They are not colorless. They take up the safranin color, safranin stain. Okay, so they appear red after that. So these are known as gram negative bacteria because they didn't take up the crystal violet stain and gram iodine. Okay, so they are known as the gram negative bacteria. Why at this step all the bacteria appear purple? Because they are false purple bacteria lying there before washing with the alcohol or acetone. Okay, after washing with the alcohol acetone, the superficial layer of purple color on these gram negative bacteria was washed off. Okay, because of that they appear colorless, and those bacteria which take up the cell, which take up this color, appears gram positive. Why? Now what is the conclusion of this procedure? Okay, what is the conclusion? So let me write down the conclusion now. Let me take this. Okay, so you can see that in gram positive bacteria, the stain was taken up by the cell wall made up of peptidoglycan. Okay, and thus it is stained the gram positive bacteria. In gram positive bacteria, the crystal violet stain and the gram iodine was taken up by this peptidoglycan. Okay, it reached at this cell wall and this is taken up by the peptidoglycan, and thus it appears purple in color after staining. Okay, but why doesn't in gram negative bacteria? Because this cell wall also has peptidoglycan. Okay, there is so why it doesn't take up the purple color? It's because, but we can say that in gram negative bacteria the stain doesn't reach the cell wall due to the presence of this extra layer on the cell wall, that is glycoproteinaceous layer. Okay, so because of that the stain doesn't reach the this cell wall, and because of that it doesn't get stained. When we stain the gram negative bacteria with safranin, the stain was taken up by this glycoprotein layer. Okay, this glycoprotein layer take up the stain. Because of that it appears red in color. Okay, you know, gram negative bacteria are more harmful to us because of the because of this outer layer of glycoproteinaceous this outer glycoproteinaceous layer. Okay, why? Because the drugs, the medicines we use to treat the disease has to cross this multi-layer system, and thus they are hard to kill. And mostly disease causing bacteria are gram negative bacteria like Vibrio cholerae. Okay, so these are gram negative bacteria which cause most serious diseases, and as well as they are hard to kill because of the outer layer, this outer layer, because of this outer layer. Okay, so this is about gram staining. I hope you get understand here. Okay, if you have any doubt, let me know in the comment box. Sorry. Okay, so if you like the video, please hit the like button and also subscribe the channel. To get the latest videos and latest update, click the bell button as well. Thank you and stay safe.